Hello everyone and welcome back to our new video. So in this video that we are going to discuss about some MCQs on the topic plant tissue culture. So this would be our part 1. Moving to our first question, what is the primary purpose of plant tissue culture? Option A, to produce genetically modified plants. Option B, to study plant growth and development. Option C, to propagate plant rapidly. And option D, to control plant diseases. The correct answer for this question is option C, to propagate plants rapidly. Coming to the second question, which plant tissue is commonly used as an explant in tissue culture? Option A, root tissues. Option B, stem tissues. Option C, leaf tissues. And option D, flower tissues. So the answer for this question is option B, which is stem tissues. So the stem tissues is commonly used as an explant for normally tissue culture purposes. Coming to the third question, what is the role of cytokinins in plant tissue culture? So let's look for the options. Option A, promote root growth. Option B, stimulate shoot formation. Option C, inhibit cell division. And option D, enhance leaf development. So the correct answer for this question is option B, which is stimulate shoot formation. So let's see our fourth question. Which hormone is used to induce callus formation in tissue culture? Options are A. Auxin, B. Cytokinin, C. Gibberellin, D. Abscisic acid. So the correct answer for this question is option A, which is auxin. So moving on to the fifth question. What is the purpose of subculture in plant tissue culture? Options are option A, to transfer plants to soil. Option B, to maintain cultures over time. Option C, to induce flowering. And option D, to study plant anatomy. I know this as a bit easy question. So the correct answer for this question is option B, to maintain cultures over time. So next we are moving to the next question. Which technique is used for the micropropagation of plant in tissue culture? Option A, grafting. Option B, somatic hybridization. Option C, embryo culture. And option D, meristem culture. So the answer for the question is option D, which is meristem culture. Coming to the next question, what is the significance of plant tissue culture in agriculture? Options. Option A, increases genetic diversity. Option B, accelerates plant breeding. Option C, reduces environmental impact. And option D, enhances soil fertility. The correct answer for this question is option B, which is accelerates plant breeding. So, coming to the eighth question, what is the term for the process of transferring plantlets from culture vessels to soil called? So, here are the options. Option A, acclimatization. Option B, transplantation. Option C, adaptation. And option D, segregation. So, the correct answer for this question is option A, which is acclimatization. So, coming to next question, which type of culture is used for preserving plant germplasm for long-term storage? So the options are option A, suspension culture, option B, meristem culture, option C, callus culture, and option D, embryo culture. The correct answer for this question is option A, which is suspension culture. So coming to our very last question in this part 1. So our 10th question is, which technique is used for the regeneration of whole plants from single cells or protoplast? And the options are option E, somatic embryogenesis, option B, somatic hybridization, option C, somatic mutation, and option D, somatic fusion. So the correct answer for this question is option A, which is somatic embryogenesis. That's all about today's video. Thank you so much for your watching. If you like this video, please subscribe to my channel.